Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive into the data, a quick but crucial ask. If you find this analysis valuable, please take a moment to hit that like button, subscribe, and share this information with your friends and family. Your support truly enables the Science Experience to continue exploring complex topics like the one we have today. Today, we are undertaking a deep dive into, well, the ticking clock of the human body, specifically focusing on the brain. Aging is the single largest risk factor for almost every major disease, from cancer to dementia. It makes it an urgent global health priority, really. And while we all track our chronological age, how many birthdays we've had, researchers are obsessed with biological age, how old your body truly is at a cellular level.
That's exactly right. And we're focusing today on a really refined biological marker that comes from neuroimaging, the brain age gap, or BAG, as it's often called. This isn't just a vague estimate, it's a quantitative measure. It looks at your biological brain age using MRI scans and then combines that with some pretty powerful machine learning algorithms.
Okay, so the concept sounds simple enough on the surface. The algorithm predicts a brain age, and you compare that to the person's actual chronological age. So if the scan says your brain looks, 80, but you're only 65 chronologically, that's a positive brain age gap. And what do the studies consistently tell us about having that positive gap?
Yeah, a positive BAG, basically, accelerated brain aging, it's a serious warning sign. The literature consistently links it to poorer outcomes across the board. Things like reduced cognitive function, frailty, and critically, a heightened risk for conditions like Alzheimer's disease and vascular dementia. It really seems to be an integrated biomarker reflecting systemic decline.
Right. So our mission today is to kind of unpack the genetic blueprint, the genetics underlying this acceleration. We're looking at the largest genome-wide association study, or GWAS, of this brain age gap to date. We really want to know what specific genes are involved in controlling the speed of the brain's clock. And maybe even more importantly, can we establish a causal link between lifestyle factors and this BAG?
The scale is what really changed the game here. Previous GWAS studies for BAG they were more limited in size. This new analysis pooled genetic data from up to 56,348 individuals. That's just a massive increase, about 79% more people than previous efforts. And that gives you the statistical power you need to find these more subtle genetic associations.
And with that kind of power, what did they find about heritability? Like how much of this brain aging speed is actually down to our genes?
They confirmed BAG is highly heritable. Common genetic variants, the ones shared widely in the population, account for a pretty substantial chunk of the variants. We're talking heritability estimates ranging from 23% up to 29%.
Wow, so almost a third potentially of the variation in how fast your brain ages is explained just by the genes you inherit.
Exactly. And that really solidifies the rationale for hunting down those specific genetic drivers.
Okay, so let's unpack that genetic architecture then. With that sample size, they must have found quite a few genetic loci, those specific spots on our chromosomes linked to this accelerated aging.
They certainly did. The study successfully pinpointed 59 associated loci across the genome.
59?
Yeah, and get this. 39 of those 59 loci are entirely novel discoveries. They hadn't been linked to BAG before.
39 new ones. That really underscores that this isn't about one single brain aging gene, is it?
Not at all. It shows that accelerated brain aging is driven by a highly complex, sort of distributed network of genetic variants. These variants interact across various biological systems. It's polygenic.
Okay, let's maybe start with the genes we might expect to see, the ones already known to be tied to neurodegeneration. Did the strongest signals line up with those usual suspects?
Absolutely. The single strongest association they found was at a locus known as 17q21.31, and the gene prioritized there is MAPT.
And MAPT, just to remind everyone, is the gene that codes for the tau protein, right?
Correct. Tau protein is, the core component of those neurofibrillary tangles that are one of the hallmarks of Alzheimer's disease pathology. Finding MAPT as the #1 genetic signal for BAG is a huge validation for the measure itself. It strongly suggests that the biological tendency to accumulate key Alzheimer's hallmarks is actually reflected in this broader measure of accelerated brain aging, potentially years before symptoms show up.
It's like seeing the shadow of future Alzheimer's risk reflected in the brain age score today. That's quite something.
Precisely. And they also confirmed another big one, the well-known Alzheimer's risk gene, APOE. Specifically, the famous epsilon-44 allele of APOE is a major risk factor, and this study confirmed the specific genetic variant driving that, RS429358, as a likely causal contributor to a higher or older-looking brain age gap.
Okay, so the heavy hitters of neurodegeneration are definitely represented. But what about those 39 novel loci? Any standouts there that are offering completely new ideas about what drives brain aging?
Yeah, there were definitely some intriguing new candidates. Two in particular offer really fascinating new directions for research. The first one is a gene called DPF3.
DPF3. What's known about that one?
Well, DPF3 has been implicated in neurogenesis and neurodevelopment, basically how the brain grows, wires itself up, and organizes itself, particularly early in life.
Wait a second. So if genes linked to how the brain is built early on are showing up as key drivers of accelerated aging later, does that kind of shift our perspective, suggesting maybe aging isn't just about wear and tear, but maybe about the initial structure?
That's exactly the implication, and it's a really interesting one. It suggests that someone's genetic predisposition towards a higher bag might actually influence the brain's developmental trajectory right from the start. The structural integrity and functional reserve built up early in life could dictate how resilient the brain is to stress, damage, and decay decades later. It's not just about decline, but maybe about the foundation.
Fascinating. Okay, and you mentioned a second novel gene.
Yes, the second one is TNIK. Now, this gene is already on the radar because it plays a role in biological pathways linked to the general hallmarks of aging, things that happen across the body, and also in maintaining neuronal health. What's really significant here is that TNIK is already being looked at as a potential drug target in other contexts, like cancer research. Finding this new link to brain aging specifically makes it a really interesting candidate for potential interventions aimed directly at slowing down that brain clock.
Right, that moves us from just observing links towards actual potential therapeutic targets, which is, well, that's huge. Okay, let's zoom out a bit from individual genes. When they looked at all 59 loci together using gene set enrichment analysis, what fundamental biological processes seem to be highlighted?
The analysis pointed strongly towards three critical areas. First, perhaps unsurprisingly, given recent research, was immune system-related processes, particularly involving the MHC protein complex. This really underscores the growing idea that chronic, low-grade inflammation, sometimes called inflammaging, isn't just a bystander, but plays a central role in accelerating brain aging.
So basically, an overactive or malfunctioning immune system could literally be speeding up the aging process in the brain. What was the second pathway?
The second area involved processes related to neurogenesis, which includes things like the regulation of axon extension, how neurons reach out and connect. This ties back nicely to that DPF3 gene we mentioned and the idea that the brain's ability perhaps to repair itself, grow new connections, or even just maintain the existing ones might be impaired in people with a higher bag.
Okay, immune function, neurogenesis.
Yeah.
And the third one sounded quite technical. Small GT-pace binding. Can you break that down a bit? What do these proteins do?
Yeah, it sounds complex, but think of small GTPase binding proteins as really crucial signaling switches inside our cells. They act almost like biological timers or regulators. They manage fundamental processes, things like cell growth, differentiation, movement, how cells respond to signals. Finding them enriched here suggests that accelerated brain aging might not just be about structural failure, but maybe a more general slowdown or error in the basic operational timing and communication within the brain cells themselves.
That really makes the brain age gap sound like it's reflecting the health of the whole system, not just the brain tissue itself. So let's connect this back to, well, real life. How did the researchers make sure this bag measure is actually relevant to people's overall health?
Right, the validation step. They compared the bag estimates against data on over 7,000 different health traits or phenotypes in the study participants, and the results were pretty stark. Higher bag scores, meaning that accelerated brain aging, correlated strongly with known unhealthy factors and outcomes. Specific examples included things like a higher number of pack years of smoking, higher diastolic blood pressure, that's the DBP, having a diagnosis of type 2 diabetes, and also, importantly, poor performance on cognitive tests, like the symbol digit matching test.
Okay, so that definitely sounds like strong evidence that a high bag is kind of summarizing poor metabolic and vascular health and projecting that onto the brain's apparent age. But as we always say, correlation isn't causation, right?
Exactly. And that's the critical next step they took in this study. They moved beyond just observing correlations to trying to establish causality using a technique called Mendelian randomization, or
Yeah,
MIFR.
right. Mesarcum. Could you quickly explain for our listeners why Mendelian randomization is such a powerful tool in this kind of research?
Sure, MR uses genetic variants as kind of a natural experiment. Since the genes you inherit are essentially randomized at conception, like a genetic lottery, we can use variants known to influence a risk factor, like blood pressure, as a proxy for that risk factor. Because your genes come first, before lifestyle choices or disease development, this method helps get around some of the biases in purely observational studies. It lets us ask, if you were genetically predisposed to, say, higher blood pressure, does that cause you to have a higher BAG? It helps infer causality.
Okay, so applying this rigorous causal analysis, what did they find? Did any of these correlated factors actually seem to cause faster brain aging?
Yes. Crucially, the MR analysis pointed towards a causal role for two major and importantly modifiable risk factors in driving accelerated brain aging. First up was high blood pressure, and this included both diastolic DBP and systolic SBP blood pressure.
A causal link. How significant was the effect?
It was quite significant. The analysis suggested that a one standard deviation increase in blood pressure causally contributes to approximately a 0.5-year increase in the brain age gap. Half a year older brain age causally linked to that rise in blood pressure.
Half a year. Just from that jump in blood pressure, that feels like a really concrete, actionable finding. Does it point to direct damage, or is BP maybe a proxy for overall vascular health?
It strongly confirms the central role of vascular health. That half-year figure is powerful for clinicians and patients alike. It really suggests that managing hypertension isn't just about protecting your heart. It seems to be directly slowing down the aging clock in your brain, too.
Okay, that's blood pressure. What was the second major causal factor identified?
The second one was type 2 diabetes. It also showed a significant causal effect on accelerating the brain age gap. So the big takeaway here is that BAG isn't just passively reflecting some inevitable neurodegenerative process decades down the line. It seems to be acting as a much more dynamic, integrated biomarker. It picks up signals from neurodevelopment, from immune function, lifestyle factors like smoking, and, as the MR showed, causally incorporates the impact of vascular health, like blood pressure, and metabolic health, like diabetes.
So this research really refines the whole picture. It maps out the genetic architecture of the brain age gap, but also highlights its incredible utility as a biomarker, reflecting this wide spectrum of systemic health issues, alongside susceptibility to diseases like Alzheimer's.
Exactly. It shows that by digging into the genetics of BAG, we get closer to understanding the underlying biology and potentially finding those levers. Maybe lifestyle changes, maybe drugs that we could use to help keep the brain biologically younger for longer.
Which brings me to a final kind of provocative thought for you based on the paper. They analyzed the genetic complexity of BAG, its polygenicity, and found it's relatively low compared to some other complex traits like neuroticism or even height. What does that suggest for the future of BAG research, say, in the next 5 years?
That's a really interesting point. It suggests we might be on the cusp of a really rapid phase of discovery for BAG genetics. While it's complex, its lower polygenicity compared to some traits means we've likely already found a good chunk of the most important high-impact genetic variants with current sample sizes. The researchers actually estimate that future studies, especially as they get really big, approaching sample sizes of maybe 1 million individuals, could rapidly identify something like 80% of the remaining unknown heritability. We could potentially map out nearly all the relevant genetic variants relatively soon. And that kind of near-complete genetic picture is exactly what you need to really pinpoint specific pathways and ultimately potential drug targets.
That is an exciting prospect. It sounds like the genetics of the brain's clock are complex, sure, but we're learning to read that code much faster than we might have thought. Well, thank you for joining us for this deep dive into the genetics of brain aging. If you found this discussion valuable, please do remember to like, subscribe, and share this information with friends and family. And thank you for supporting the Science Experience. We'll see you next time.